You're listening to Making Waves, fresh ideas in freshwater science. Making Waves is an online science podcast where we talk about innovation and discovery in freshwater research and why it matters to you. This podcast is made possible with support from the Society for Freshwater Science. Thank you for tuning in today. I'm your host, Tim Cline. Joining me is Dr. Jill Welter from St. Catherine University. She will be telling us about some research she and her colleagues conducted that was recently published in Ecology, looking at how nitrogen fixation and primary productivity may be influenced by changing temperatures. When I think about climate change and carbon cycling, and I read the headlines of major science journals, there seems to be an abundance of literature looking at how climate change is going to influence bacterial respiration and how this is going to alter carbon cycling. However, I have not really seen anything about how changing temperatures might influence rates of primary productivity. So you and your colleagues looked at this question and found perhaps an unconventional mechanism. Could you tell us about where the interest in this question came from?
Sure. Well, there are an increasing number of ecologists and also an increasing body of literature about metabolic theory of ecology, which discusses how biological processes will change with temperature. And we've known for a very long time that almost any biological process that you can think of will increase, rates will increase with temperature. So we can describe that, how that process rate should change with temperature is based on basic enzyme kinetics. So that's true for cellular respiration, as you mentioned, but it's also true for the suite of enzymes involved in photosynthesis and also any other biological mechanism that's mediated by enzymes responds exponentially to an increase in temperature. So many ecologists are studying this, and there really are a growing number of papers that describe how biological activity is changing, but it's much more complicated to study in an intact ecosystem where you have a suite of species all with, different suites of enzymes all responding to temperature in different ways. And that is one of the challenges that ecologists face, is really understanding how, not just how a single species or one particular suite of enzymes will respond to climate change, but how ecosystems as a whole, including individual organisms, populations, species interactions, and food web dynamics will respond as climate warms. And our particular work in Iceland has focused on those questions, but I have really come in on the team as I've been really interested in nitrogen fixation and how it fits into this story. Because often, even when we think about metabolic theory of ecology and how biological process rates will respond to warming, there's been a lot less work to understand how changes in resource supply may feed back on responses to change in temperature. And so, for example, there are many processes, including chemical weathering, but also biologically mediated processes like biological nitrogen fixation that also process rates increase with temperature. And so as we see a warming world, we may increase the availability of limiting resources like nitrogen in some ecosystems. And as we do so, how is that going to change our predictions about how processes like gross primary production and cellular respiration may respond now that there is higher availability of this limiting resource?
Not all of our listeners are likely to be intimately familiar with nitrogen fixation. Could you provide a basic primer on nitrogen fixation and how temperature influences this process?
Yeah, nitrogen fixation is really an exciting process, and it's one that most ecosystem scientists really haven't paid much attention to, especially in freshwater ecosystems. Those of us who do study nutrient cycling tend to focus on certain transformations in the nitrogen cycle, but there hasn't been as much attention paid to nitrogen fixation. But it is experiencing a bit of a revival right now, that there are more and more people interested in nitrogen fixation, and they can see the important role that it is playing in freshwater ecosystems in particular. And so nitrogen fixation is a process that's completed by a group of organisms cyanobacteria, and my work focuses primarily on photosynthetic or autotrophic nitrogen fixers, but there are many ecologists that also study heterotrophic nitrogen fixers. And this group of organisms is capable of accessing nitrogen gas from the atmosphere as a source of nitrogen for growth. And so the enzyme that's required for this process is called nitrogenase. So this is an enzyme-mediated transformation of nitrogen. It actually requires quite a bit of cellular energy to break that triple bond in nitrogen gas and then to be able to use that nitrogen for their own growth. So in ecosystems, particularly those that are nitrogen limited, nitrogen-fixing organisms have a great advantage in that they can still access nitrogen from the atmosphere, whereas other organisms that aren't capable of this process continue to be nitrogen limited and experience low availability. So in the context of our work, which it happens to be in the Hengil region of Iceland, which is a very nitrogen-poor landscape, that nitrogen-fixing cyanobacteria, primarily several species of nostoc, really take hold in some of these rivers. They're very nitrogen-poor, so these nitrogen-fixing organisms have a real advantage. And one question is, how is the presence of these nitrogen-fixing species likely to influence the response of rivers and lakes to increasing temperature? Because they do bring a limiting resource into the ecosystem. And since this is an enzyme-mediated process, the rate of nitrogen fixation also increases exponentially with temperature. And nitrogen fixation in particular, or the nitrogenase enzyme, has a very strong temperature dependence. So rates of nitrogen fixation increase dramatically with each step temperature increase. So with warming, we expect to see big increases in nitrogen fixation, which means a big increase in the amount of nitrogen that's entering that ecosystem. So how is that then, that increase in supply of that limiting resource, going to feed back on gross parent production and respiration and help better inform climate change predictions for freshwater ecosystems?
So I understand that you set out to look at these issues using a combined field study and manipulation. Could you describe how you designed the study to test these questions?
You know, we really are so lucky to have such a fantastic field site where we can test many of these ideas. You know, we're working with an international team of ecologists in Iceland. The work is focused in the Hengil region of Iceland, where we can literally walk about 45 minutes and cross 18 streams that vary by up to 25 degrees Celsius in temperature. And it's just such a great natural experiment. So we have all spring-fed rivers that have largely the same solute chemistry, the same aspect, similar flow velocities, but differ in temperature. So you couldn't have imagined a better landscape to ask questions about how temperature will influence river ecosystems. And so we've been lucky to work there with the Institute for Freshwater Fisheries and the University of Iceland in this collaboration that is still ongoing. And so we've completed some experiments in the landscape streams themselves that vary in temperature. And then last summer, we also set up an experiment where we piped water from a cold stream through a series of heat exchangers, or basically metal tubing that we placed in some hot pots to warm it up, and moved that water into a series of coolers where we could manipulate water temperature just by basically creating a cold faucet from the cold stream and a warm faucet from the water that was warmed up as it passed through the piping in the hot pots. And we created an experiment where we piped this water into a series of experimental channels to create 5 different temperature treatments. And I should give a shout out to Tanner Williamson, who's a master's student who was really in charge of this project and did all of the hard work to maintain it every day and make sure the temperature regimes were as they should be, and make sure the flows were as they should be, and just kept an eye on everything. And he just completed his master's degree at Montana State with Wyatt Cross. So what we were interested to see as we move across the temperature gradient in the experimental channels, how primarily gross primary production, respiration, and nitrogen fixation rates changed with increasing temperature. And we have expected relationships that come from metabolic theory. So this was a really nice design in that we could compare our empirical estimates of these processes and how they change with temperature to published values that are based on enzyme kinetics for cellular processes, including gross primary production, respiration, and nitrogen fixation. So we completed the experiment, and what we found in terms of nitrogen fixation is that rates increased dramatically across the temperature gradient, and were very similar to what the temperature relationship or temperature dependence of nitrogen fixation that we would expect when we look in the literature. So first, in terms of our results for nitrogen fixation, as I mentioned, we had 5 temperature treatments. We had 5 temperature treatments, beginning with ambient stream temperature, and then 4 temperature treatments that were roughly increasing by 5-degree increments. So we spanned a temperature gradient from about 7.5 to 23.5 degrees Celsius. And our results in terms of nitrogen fixation, across that temperature gradient, we did see dramatic increases in nitrogen fixation. In fact, nitrogen fixation rates increased 71-fold across that temperature gradient. So we saw high temperature dependence of nitrogen fixation, and we also saw that overall the rate of increase in nitrogen fixation matched what you see in the literature for the nitrogenase enzyme. So increase in our process rates in these biofilm communities very closely matched what we see when you isolate the nitrogenase enzyme in the laboratory. So it appears to be very much controlled by enzyme kinetics, and our field experiment matched that prediction very well. But we saw a different story when we took a look at gross primary production and cellular respiration. We found that for both processes, the increase in process rate with increasing temperature was much more dramatic or amplified over what you would expect when you look at rates in the literature just based on subcellular processes. So a much more amplified increase. In fact, let me give you the numbers. Gross primary production increased 53-fold across the temperature gradient, and cellular respiration increased 32-fold across the temperature gradient. And a much higher temperature dependence or amplified rates of increase across that gradient than you would expect from the literature. And in fact, when we took a look at the shapes of those relationships, we found that the increases in gross primary production and cellular respiration mapped very well onto the pattern we saw for nitrogen fixation, which suggested to us that these amplified temperature dependences we observed in gross primary production and respiration were likely driven by the temperature dependence of nitrogen fixation, or the amplified rates of gross primary production and respiration were driven by the increased nitrogen supply associated with nitrogen fixation with warming.
So these are very striking results. What do you think the big takeaway messages are, or the broader importance of this research for understanding ecosystems and environmental change?
In the end, the results of our experiment aren't that surprising. If you increase the supply of a limiting resource, you expect the ecosystem to respond. You expect to see increased rates of growth, a response of gross primary production and respiration to that increased availability of a limiting resource. So that in and of itself isn't that surprising. But it is important that we begin to couple the temperature dependence of these different processes so that we can generate better climate change predictions for a variety of ecosystems. And that has one aspect that's been missing from metabolic theory models. And so I think that's a really important direction in which we are currently moving, but we certainly need more work there. Also the amplification of the temperature dependence of gross primary production could be fairly widespread under climate warming. especially in ecosystems that will respond to added nitrogen that may be supplied by increased rates of nitrogen fixation. And so I think that's a question that remains as well. Where and when may nitrogen fixers, increase rates of activity and increase biomass in ways that begins to feed back on the response of the ecosystem to warming climate. And that's an area where we don't have much information and could be potentially really important. There are a lot of food web implications here as well that certainly feed back on ecosystem metabolism in interesting ways. Under climate warming, we may see, more dramatic shifts in the base of the aquatic food web from a biofilm that is more dominated by species of green algae and diatoms that are highly edible and good sources of food for invertebrates towards less edible, less palatable cyanobacteria. Therefore, that can shift the nitrogen that's available in those organisms may not be available to fuel higher trophic levels, may not be transferred into the food web, but instead exported downstream. This has huge implications, right? If the base of the food web is edible and supports the food web, you have one trajectory for nitrogen through the ecosystem. If the food is inedible and full of nitrogen, you have a completely different trajectory. It's going to be downstream export, to be utilized at some point downstream. But that really changes how nutrients are being cycled and the functioning of the ecosystem.
This podcast was brought to you with support from the Society for Freshwater Science. For more information about this speaker, the podcast, or the Society, please visit www.freshwater-science.org. Be sure to join us each month for more fresh ideas in freshwater science. Thanks for listening.